This is an animation depicting the ultrastructure of a sperm. Before starting with the explanation of the structure, I would like to tell you a few important facts about sperms. Sperm cells are gametes that are produced in the gonad of male human beings. They carry a total of 23 chromosomes. Human sperms are approximately 60 microns long and have an average lifespan of 3 to 5 days. Now the question arises that how the sperms are formed and matured. So, in the inner walls of the seminiferous tubules of testis, spermatogonia undergo a mitotic division to produce primary haploid spermatocytes. These cells then undergo meiotic division, resulting in the production of secondary spermatocytes. The spermatids are formed after spermatocytes undergo the second meiotic division. This spermatogenesis takes about 74 days to complete. Mature sperm cells, known as spermatozoon, are formed from spermatids after a process called spermiogenesis. Now, as we finish with this basic stuff that we needed to know, we can finally begin with the structure of the sperm. The mature spermatozoon can be broadly divided into head, neck, middle piece, and tail. The head of the human spermatozoon is oval on surface view and pear-shaped on the side view as shown in the animation. It is elastic and measures about 4 to 5 micrometers in length. The head is a condensed nucleus and contains about 40% of DNA and is rich in arginine. The genetic material is contained inside the nuclear envelope, and the nuclear pores can also be seen in the animation. The sperm can be seen covered by plasmalemma that has also been talked about later in this video. The acrosome is formed out of the Golgi apparatus of the spermatid and can liberate chemical substance which enables the sperm to pierce the zona pellucida of the ovum. As the animation zooms in on the next part of the sperm, we can see the neck as a short, weak segment that connects the head with the middle piece, also known as the body. The proximal centriole fits in the depression in the head and it is the junction of the head and neck, whereas distal centriole lies between the neck and the middle piece. The next portion of the video focuses on the middle piece or body of the sperm. In the middle piece, the helical mitochondrial sheath consists of 10 to 14 turns in which mitochondrial cisternae are found to be fused with its limiting double membrane with lipid-rich substance. The axial fiber bundle is the central filamentous body which begins from the proximal centriole and ends in the end piece that is marked as dense fibers in this video. Except in the end piece, the axial filaments are surrounded by the axial sheath, which is formed out of condensation of a thin layer of cytoplasm. You can see the 360 degrees view of the middle piece in this frame. Now the tail of the sperm can be seen, that is labeled with its fibrous sheaths and the flagellum. You can once again see the axial filaments that had begun from the proximal centriole and can be seen elongating all through the end piece. The longitudinal filaments are surrounded by a fibrous sheath of circumferentially oriented dense fibers. The tail is about 45 micrometers long and about half a micrometer thick at the base, gradually tapering towards the end piece. ATPase enzyme is present throughout the whole length of the tail and helps in the movement of the spermatozoon. A little fact about the semen is that there are about 100,000 sperms per cubic millimeter in healthy males ejaculate. On the ending note, you can again visualize the sperm in its entirety and appreciate the structures in every plane.